He then completed his MTech in Biological Sciences and Bioengineering from the Indian Institute of Technology, Kanpur in 2010. He completed his doctoral dissertation work at the Academy of Scientific and Innovative Research, Central Scientific Instruments Organization, Chandigarh, under the Council of Scientific and Industrial Research, CSIR, India. Since January 2012, he has been working as a scientist at CSIR under the Central Scientific Instruments Organization, CSIO, Chandigarh, India. His current research interest includes the development of nanomaterials such as fluorescent semiconductor quantum dots, carbon dots, and gold nanoparticles for biosensing applications. He is also developing a highly porous metal organic framework, also known as MOF or MOFs, as a platform for stimuli responsive drug delivery and antimicrobial agents. In recent years, he has also started working in the area of optical sensors for chemical and biological sensing. Now over to you, Mohanta sir.
Am I audible now?
Yes, sir, you're audible.
Yeah, thank you for wonderful introduction. Let me just share my slides. Tell me if they are visible or not. Okay. Is it visible?
Yes, sir, it's visible.
Oh, yes. Let me put in presentation mode. Is it in the slideshow mode? Slideshow mode?
Yes, sir, you may continue.
Okay. Okay. Thank you. So basically, for this field of carbon quantum dots, I have done some work over this for biosensing applications, and in since I did not know what is the general audience that I'm going to present this, so I have kept it a little general talk just to know about kind of introduction to the carbon quantum dots. So now going before to carbon quantum dots, let us just recall what we have learned about nanomaterials, because carbon quantum dots are a kind of a nanomaterial. So what happens is that these nanomaterials, they are having any one of their dimension in below 100 nanometer range. So any material which is having there at least one of the dimension in below 100 nanometer range, they can be called as nanomaterials. So based on that, they could be divided as 2D materials, where one of the dimension is below 100 nanometer, or they could be one dimensional nanomaterials. For example, nanowires are there, nanoribbons are there, and they could be zero dimensional nanomaterials. Now, these nanomaterials, when we compress any material to this nano size level, to the nanometer scale range, there are two prominent effects that comes into play, which is otherwise not present in the bulk materials of the same material. It is not present in the bulk counterpart, but it becomes dominant when we go to the nanometer range. So these two effects are, one is surface effect and another is the quantum confinement effect. I am not a physicist, so I will not be able to go to a very in-depth analysis of these effects. But in general, the overall concept and how they affect that can be easily understood by anyone. For example, in the surface effects, now consider this is a cube. Okay, so this cube has a 3 sides, and if we calculate the surface area, it will come around area of that particular face multiplied by, there are 6 faces, so around 54. And the volume of the cube is obviously a cube, a side cube, so it is 27. Now the same cube, when it is divided into smaller and smaller portion, what we observe that their surface to volume ratio tremendously increases. So for a particular material, when we go down the size to the nanometer level, what happens? The surface effects becomes prominent, because now number of the percentage of atoms which are present at the surface is very high. And these surface atoms, now mostly these surface atoms are uncoordinated, because from the if let's say if a atom has a tendency to form 8 bonds, but those atoms which are present at the surface have neighboring atoms just below them. So therefore, or the atoms which are at the edges, they have neighboring atoms, just a very smaller number of neighboring atoms. So these uncoordinated atoms which are present at the surface, they are very highly reactive. And because of this very high surface to volume ratio in nanomaterials, their property completely changes as compared to their bulk counterpart. For they have enhanced interaction with their surrounding medium. Therefore, if any material is optically active, it will have a large absorbance uh, when we go down to nanometer scale. For we can easily recall the example of gold here. Like the bulk gold does not absorb light in the visible range. It absorbs in the so but whereas when you go to gold nanoparticles, we get a prominent peak of absorption at around 520 to 550 nanometer, depending on the size of the nanoparticle. So there is a tremendous change in the optical property of the material when we go to the nanometer level. Then there is chemical property changes also. For example, the same gold nanoparticle, or the gold which we consider as very inert, and therefore we use it as jewelry and all these stuffs where it does not get corroded with time. But the same gold becomes very active and have been shown to very catalytically active at the nanometer range. There, the melting point of a particular metal, when it is taken down to a nanometer range, it also decreases. So these are the various physical and optical and electronic property changes. There are further changes also. For example, hello.
Sorry, sir, there have been a small
Okay.
disturbance. Continue.
No issue, no issue. So we see changes when we take iron. So when we have iron, it is ferromagnetic in a bulk composition. But if we take down to nanomaterial scale, it becomes super paramagnetic. What does it mean is that the super paramagnetic the nanoparticles of iron, they do not possess as such magnetic activity. But when we bring any magnet to near to them, they are quickly attracted to it. The another effect that I was talking about was the quantum confinement effect. As I've previously told you, I'm not from the physics background. So for the detailed physics, the quantum physics of this, you can refer to other books as well. But in general, what happens is that, let's say if we have a molecule, and these molecules are made up by combining atoms. So these atoms have some half-filled orbitals, or empty orbitals, atomic orbitals, or filled orbitals. So what happens that when these atoms combine into a molecule, they form molecular orbitals which are filled and some are unfilled. So this unfilled, they goes to LUMO, that is they goes to uh, uh, LUMO, and the filled orbitals, they forms to HOMO. And the difference between the highest filled orbital, highest filled orbital, and the lowest unoccupied orbital, it becomes the band gap for that material. So these orbitals, they occupy a particular energy states, and the difference between them is the band gap energy for that material. And when the multiple molecular orbitals combine with each other, they forms a continuous conduction band and the valence band that we normally call. What happens, particularly in semiconductor nanomaterial, when we go from molecular level to cluster level to nanoparticle of different sizes, there is a shift in the band gap energy, and based on the size, the band gap energy changes. And when we go from lower size to higher size, the band gap energy decreases, and therefore the energy which is required by electron to jump from the valence band to the conduction band also decreases accordingly. Now, since there is no in in the bulk counterpart, there is a continuous formation of energy bands, that like there are all the available energy states where electron can go. But in case of nanoparticles, what happens when electron is excited, it has a very limited set of energy states where it could actually go because of this confinement, because there are not much energy states available to the electron to go and release its energy. It has to come down, it wants to come down, release its energy, but there are not many available states. So it will go to only to the very finite number of available states, and because of this confinement, it majorly loses its energy as a radiative power. That means it loses energy with radiation, with emission. So by just tuning this size of the nanomaterial, we can therefore tune the band gap of the semiconductor material, as well as we can tune the optical emission that we will be getting out from this material. So now coming to carbon. So these two effects which I have discussed earlier, this is also observed in carbon. But if we first consider the bulk allotropes of carbon, there are two. One is graphite and the diamond. These are the three-dimensional bulk allotropes of carbon. What happens? We can easily see that in the graphite, the carbons are sp2 hybridized. That means each carbon is bonded with another 3 carbon atoms. Whereas in diamond, a carbon is hybridized with 4 other carbon atoms and taking a sp3 hybridization, which gives it a tetrahedral shape. Just with the difference of this sp2 and sp3 hybridization, we can see a tremendous change in the physical properties of these two materials. Like we all know that diamond is a very transparent material and it does not conduct electricity. It is a hardest material that we know of. Whereas in case of graphite, it's a very good conductor of electricity because of the free electron now present, and it is very brittle material. We can easily break it. The pencil lead that we are using is made up of graphite, so it is very brittle. So same carbon atom composition, the carbon composition is same, just based on the bonding between the atomic bonding or the atomic network between the carbon atoms, we get a very vastly different properties for that material. So this we have to keep in mind when we go further ahead. Coming to the carbon nanostructures, the scientists have identified majorly 5 carbon allotropes or carbon nanoallotropes. These are carbon nanodiamonds, carbon fullerenes, carbon dots that we are talking today about, carbon nanotubes, and graphene. The graphene represents the two-dimensional allotrope for carbon, where all the carbons are sp2 hybridized and they are arranged in a linear fashion. So they are we can consider them bulk in xy direction, but in the z direction they are in a nanometric scale. Similarly, carbon nanotubes, some call it as just rolled over graphene. They could be as a single walled or multi-walled. Here also all the carbons are sp2 hybridized. Similarly, the earliest discovered carbon allotrope was carbon fullerene, where the well-known is C60 or buckyball, that we call as buckyball, where the carbon atoms, they are sp2 hybridized again, and using a network of hexagon and pentagon, they are arranged in a spherical shape. So these carbon fullerenes, carbon nanotubes, and carbon and graphene, they are all sp2 hybridized carbons, and therefore they have some similar properties. For example, all of them are very good conductor of electricity. Then using these 1D carbon nanotubes and graphene, there are several other nanostructures that people have now derived, researchers have now derived. For example, there are graphene quantum dots, which are similar to carbon dots, but their difference is that they are derived from carbon, they are derived from graphenes, and they are usually nanosheets rather than a spherical dots. Then graphene oxide is there, which is obtained by just oxidizing a graphene sheet. Then single walled CNTs are there, carbon nanotubes are there, multi-walled are there, reduced graphene oxide are there. So there are several other derivatives which have come out of this nanoallotropes. Now there is, and in the 0 dimension, there is a carbon allotrope of carbon nanodiamonds and carbon dots. Now these two are spherical in nature. They are very similar from physical characterization, but the major difference there lies with the composition of their carbon atom, which in carbon dots they are usually mixture of sp2 and sp3 hybridized carbon dots, as well as there are some amorphous carbon atoms which are not arranged in any particular fashion, and there are many heteroatoms. For example, there are nitrogen, there are oxygen, sulfur atoms also present on the carbon dots. In case of nanodiamonds, the majority of carbon dots are arranged in sp3 hybridization, and they are also called as diamondites. Like diamond like structures are there in the core of the nanodiamonds. And as compared to nanodiamonds, carbon dots are highly fluorescent, and therefore majority of research focus have always been directed towards carbon dots and not much with nanodiamonds. Another problem with nanodiamond is that they are acquired only through high combustion. They are acquired or they are produced during high combustion events. For example, some explosion is there, then only nanodiamonds are usually produced. So therefore, majority of research has been focused on carbon dots because they could be very easily produced in the laboratory conditions and in a bulk quantity. Coming to carbon dots, or which are also called as carbon quantum dots. So in this talk, I will be using them interchangeably. So whenever I say carbon quantum dots or carbon dots, please consider them as equivalent. This should be carbon dots. I'm sorry, spelling mistake is there. Carbon dots. So this is the first paper where the carbon dots were actually discovered in a by chance fashion. What happened is that these authors, Zhu and Scrivens, they were working with single walled carbon nanotubes. So during the purification of single walled nanotubes, what they did electrophoresis, the general electrophoresis that we do in our laboratories, in 1% agarose gel, and when they observed this agarose gel under UV illumination, they observed that there is another band of highly fluorescent material. They did not name it carbon dots then. They just call it single walled carbon nanotube fragments because they considered that these are derived from the single walled carbon nanotubes. So these highly fluorescent nanobands, when they studied it further, they found it that it could be eluted in different fractions when they are doing some column purification. So that suggested these people that whatever this fluorescent material is, it is of different size. It is not of single size. So they therefore confirm the size of these nanoparticles, or now we can call this carbon dots, using atomic force microscopy, and they found out that majority of the particles were below 2 nanometer. So they established a fact that whenever carbon nanoparticles, they are in the nanometer range, around 2 nanometer or lesser, they have a very high fluorescent properties. This was not observed before. This is the first time that they observed something like this using carbon. Because CNTs and graphene at the time in 2004, they were majorly studied for their electrical properties. So no one was looking for their optical properties. So that was the serendipitous discovery here that they found. They also found the size could go up to 18 nanometer for red colored or orange colored nanoparticles. So there was size difference was there. When they studied the elemental composition for these carbon dots, they found out that these dots are entirely made up of carbon. They are completely metal free. So there is no there is no possibility of some metal complex being fluorescent there, because there are several metal complexes which are also fluorescent. For example, there are copper iodide complexes are there, which are very fluorescent when they are complexing with some organic molecules. So that was not the case. So it was completely metal free, and the composition was completely made up of carbon, hydrogen, nitrogen, and oxygen. Like heteroatoms were also there. And when they studied FTIR, these nanoparticles, carbon dots under FTIR, they found out the presence of carboxyl, hydroxyl, and several other polar functional groups. This was also very important because this gave an idea to researchers that these nanoparticles could be utilized in biomedical domain, or because of their very high water solubility, as well as their functional groups, which could be modified further. So the research now focused majorly on the fluorescence of the carbon dots. So people now were very much interested, how can we control the fluorescence of these carbon dots, because that was the main aim, right? Because it is a fluorescent material, so one should be able to control the fluorescence of this material, then only we can be able to do something meaningful out of it. So presently, the fluorescence which is generated in the carbon dot, it is still debatable. I mean, it is precise mechanism is still unknown, but there are three main effects, or we can say theories, which has come into picture and people have accepted it. First is the quantum confinement effect that I have already explained in the previous slides, where we can see when the particle size is decreasing, the band gap of the particle increases, and accordingly the fluorescence of the particle shifts from red to blue, or it shows a blue shift. So in this paper, what they did, they generated carbon dots using a very precise alkali assisted electrochemical oxidation. What they used a graphitic tip in the anode, and they passed electric current. And because of the alkali present in the electrolyte, the graphitic layers which are present on the anode, they got oxidized and they got released into the solution. And that solution, when they studied, it showed fluorescence. The carbon dot was produced, and based on the current that they are passing through the anodic tip, they could control the size of the carbon dots from 1.2 to 3.8 nanometer, and they could see that when the size is changing, there is fluorescence emission is also changing. So after this quantum confinement effect, people also studied the surface effect of carbon dots. As I discussed previously, what these people were able to show, or the researchers were able to show, that if we control the degree of surface oxidation of carbon dots, we can actually change the luminescence or the emission profile of the carbon dot. In this example, what they did is they prepared the carbon dots, which they have indicated here, having two kind of core shell structure. The core is of graphitic nature, that is all the sp2 hybridized carbons are present in the core, and the surface is present of heteroatoms like carbon, oxygen, nitrogen, and these are present in the heteroatoms. So they specifically studied the level of oxidation in this study. So what they studied, oxygen is a electron rich atom. So when oxygen is introduced into the carbon dots by successive oxidation, what they observed, that they could theoretically also study that it changes the band gap of the carbon dots
इसलिए
without
जबरदस्ती ज्वाइन करवा रहे हैं
without affecting the band gap of the particles. So they observed that when with the increasing in the oxidation state, oxidation of surface oxidation of the carbon dots, so there is a decrease in the band gap energy, and simultaneously there is a redshift in the fluorescence emission. Third theory regarding the fluorescence of the carbon dot is that, for example, this is in this study, what they did, they used citric acid and ethanolamine, and they subjected to pyrolysis at very high temperature. Pyrolysis means there is a removal of water from the carbonaceous content, so therefore it's minus H2O. And when they carried out this pyrolysis at different temperatures, one at 180 degrees Celsius, another at 230, and progressively to 300 and 400 degrees Celsius. What they observed, that when they did pyrolysis at lower temperature, so it was a very fluorescent carbon dot was produced, and they attributed this fluorescence to something called as molecular fluorophores, which are generated on the surface of the carbon dots. So again, this is a surface effect. Majority of it is a surface effect. And when we increase the temperature of pyrolysis, they could find two type of fluorescence, one emanating from the core, graphitic core of the carbon dots, and another from the surface fluorophores. And as we increase the temperature, the higher amount of surface fluorophores are converted into graphitic carbon and goes into core, and therefore at very high temperature of pyrolysis, there is a significant decrease in the fluorescence intensity, and the emission from the surface fluorophore quenches, whereas emission from the graphitic core, it's only present in very high temperature pyrolysis. So these three theory gave a picture of fluorescence control for the carbon dots, which is very important when we go further in further applications. Another very interesting way of controlling the fluorescence of the carbon dot was using doping. For example, carbon dots is a structure of carbon atoms present in a sp2 hybridized or sp3 hybridized network. So if researchers can introduce highly electronegative elements like fluorine, nitrogen, sulfur into the carbon structure, because of the change in the electron density, there will be change in the band gap of the carbon dots, which is ultimately forming. So people find out that when they produced carbon dots which are doped with fluorine, they could tune the band gap towards the red emission. So now your fluorescence goes from green to red. So your band gap energy has actually decreased because by introducing the electron rich carbon fluorine atoms, they have produced more number of energy states where your electron can now pass on the energy and comes to the ground state. Another very interesting phenomena that they found out with carbon dot is that the carbon dot can not only doped with inorganic elements like nitrogen, sulfur or oxygen, they could be also doped with metallic ions. For example, people have seen that if carbon dot is doped with zinc or cadmium, the fluorescence is shifted, as well as the quantum yield of the fluorescence also increases. If we use a lanthanide series of elements like europium or terbium, the fluorescence of carbon dot shifts to the emission of those elements, of those lanthanide ions, and it also increases. In this particular example, what people did is they doped the carbon dots with gadolinium, and now with fluorescence imaging, they could also image the carbon dots in a MRI, in a T1 weighted MRI. So because of this gadolinium, it is a paramagnetic metal ion. It gives a MRI response. So when incorporating the carbon dots with the gadolinium ion, we can get dual mode imaging, where we can image the carbon dot with fluorescence as well as with MRI response. Then there are two very unique properties which is present in carbon dots, which is usually not seen, and these two properties are usually not seen in the traditional semiconductor quantum dots like cadmium selenide or zinc selenide quantum dots. These two properties are: first, excitation dependent emission, where the carbon dot produces different emission profile when it is uh, excited with different wavelength. For example, when the carbon dot is excited with 330 nanometer, it has an emission profile of around 400 nanometer. That is blue light is emitting. And when the excitation is shifted to 460 nanometer, it produces green light. So it emits in the green region. So this excitation dependent emission is uniquely present in carbon dots. Second, second very important property of carbon dot is that since there are several functional groups that we have discussed previously on the carbon dots, and therefore the polarity of the carbon dot can be easily changed, and this polarity can be easily controlled by change in pH of the surrounding medium. So therefore almost all the carbon dots, because of their polar functional groups, they are very much you can say responsive to the pH of the vicinity, or we can also control their emission profile using the pH. The carbon dots, the solutions that people have, researchers have shown that it could be stable even up to more than 1 year, and even when they are present in the, in the sunlight, keeping in the ambient light. Now, why this is very important? Normally, all the nanoparticles, they are very surface reactive, because as I told that these nanoparticles, they have majority of atoms present on the surface, that high surface to volume ratio. So almost all the nanoparticles, including the traditional quantum dots, even the gold nanoparticles, they have a very high tendency to aggregate with time. So therefore, they are usually stored at room temp 4 degrees refrigerated, or they are stored as powder, and they are used after dissolving before then immediately used after dissolving them. But in case carbon dots, now we have seen that carbon dots with, because they're highly oxidized nanoparticles, they are rather very stable even in ambient conditions. So therefore, for any biomedical applications, the aggregation of nanoparticle is one of the very limiting challenge that people face when they use a gold nanoparticle or any other nanoparticle. So because the surface is so active, so they have a tendency to aggregate. But whereas in case of carbon dots, they have very less tendency to aggregate, and they remain stable for a very long period of time. So controlling the emission of the carbon dots was one factor that actually helped researchers to go into a various application of go into their various applications. When we think from the point of biomedical applications, the another very important factor that one must keep in mind is the surface functionalization of the carbon dots. So there are several methods of surface functionalization. I am not going to details of all of them. They could be classified as covalent modification, where we attach some functional group deliberately so that we can use it for polymerization, we can use it for silica formation, or they could be interacted with various other metal ions. Like for example, I have shown you the example of gadolinium, which could be used for magnetic resonance imaging, MRI. And we could tune the hydrophilic balance of the carbon nanodots using surface functionalization. This is useful for different applications. For example, in organic LEDs, the people are now trying to utilize carbon dots as light emitting diodes. So in that, they need to take these carbon dots into a organic solvent to protect it from oxygen, atmospheric oxygen. So for that, they could integrate this carbon dot with various hydrophobic moieties so that it could be now dispersed in an organic solvent. Then carbon dots, these surface functional groups of carbon dot, they are used in tagging with proteins for, let's say, bioimaging, targeted bioimaging, or targeted drug delivery purposes. And these surface functionalization, they can be used for this stability studies in serum, buffer, or any other pH variable mediums. Another important factor before we consider carbon dots for biomedical application is their biotoxicity. So I guess everyone knows the most accepted form of cell viability assay is the MTT assay that usually people do for assessing the toxicity of any nanomaterial, be it carbon dot, be it quantum dot, be it gold nanoparticles. This is a very accepted form of testing for toxicity of any biomaterial. The first line of toxicity study is done using MTT assay, where this MTT compound is basically reduced into a purple color producing formation crystals by mitochondrial reductase. So if the cells or organisms we are talking about, if the toxicity is produced, the cells, the microbial reductases will not be able to convert this MTT into formazole, and therefore it gives us a sense of whether cells are viable or not. So in general, the toxicity study of carbon dots has been studied immensely in vivo models as well as in cellular cell lines as well. For example, HeLa or MCF cells, they are generally non-toxic, and people have found that if the functional group of the carbon nanodots is properly done, then they could be safe even up to 1 mg per ml concentration, which is very high concentration from other terms. The toxicity of the carbon dots, what people have found out that it really depends on the surface functional group. For example, if a carbon dot is functionalized with polyethylene, which is a positively charged polymer, so it produces toxic effect in the cellular studies. Whereas if the same carbon dots are pegylated, that is, it is attached with polyethylene glycol, which is very hydrophilic polymer, it is considered as a generally as biosafe. So now, considering everything, that the carbon dots are very fluorescent in nature, we have done the surface functionalization properly so that it will not produce any toxicity because of the surface functional groups. And if we want to do some targeting applications, then we can use that surface functional group for targeting with proteins or any other receptors. Now, after doing that, we come to the biomedical application. So now there are various biomedical applications for carbon dots. I have listed some. One of the earliest bio application of carbon dot was obviously bioimaging, because they are fluorescent, so they will be used for bioimaging. So in this study, they have shown the bioimaging of HeLa as well as MCF cells. And if we could target it properly, we can also go it for in vivo imaging, bioimaging, where when carbon dots are injected in the rat, they are assimilated in the bladder and uterus area, and which could give us a fluorescence imaging. The another interesting application, as I have previously discussed, that if we prepare our carbon dots doped with gadolinium or iron metal ions, which are basically responsive to magnetic field, we can get dual imaging mode, fluorescence as well as MRI imaging mode. The very interesting study using carbon dot was done utilizing their property of pH and temperature dependent fluorescence. What they have done is that when the cancerous cells grow, they reduces the pH of the vicinity because of the generation of lactic acid. So these authors, what they did, they used blue fluorescence carbon dots, which was emitting in blue in normal pH, normal physiological pH of 7.4. But as soon as the pH decreased below 6.8 because of the acidic nature of the cancer cells, it produced green emission. So based on this pH dependent emission, one can actually use carbon dots for diagnosis of cancer also. So they have shown this study in vivo in mouse model, as well as in uh, cellular model, where they could find that with change in temperature as well as pH, the fluorescence of the carbon dot is also changing. Another effect that people have cleverly utilized is that they produced carbon dots doped with manganese ions. So they use a manganese phthalocyanine. It is a photosensitizer molecule. Photosensitizer means it gets activated by emission when it is get activated or excited by light, and in response it produces reactive oxygen species. So what people have found, that when carbon dot is made with such an uh, complex, photosensitive complex, what happens is that it is not only able to produce fluorescence, but it is also able to produce uh, this photoreactive oxygen species. So in this study, what they did, they produced carbon dots using manganese complex. Manganese is paramagnetic, therefore it also shows MRI response. The carbon dot as such itself is fluorescent, so it is showing fluorescence imaging as well as. And because of the manganese complex that they used for preparation, the manganese complex is now able to, when irradiated with light, it is able to generate reactive oxygen species, and which will produce toxicity in the cancerous cells if it is targeted properly. So photodynamic therapy is another very interesting applications which which researchers have now coming up with carbon dots. On the similar line, what researchers have found is that these carbon dots, they could be doped with nitrogen or oxygen or other heteroatoms, And because of this doping, we can shift the excitation and emission of this carbon dots to the near infrared region. In the near infrared region, when these carbon dots are irradiated with the light, they are produced to generate in the local vicinity of the carbon dot. They are able to produce heat. This effect of generating heat from the photoexcitation or the excitation by the light is called as photothermal effect. So when this photothermal effect, these carbon dots, the surface functional groups, they are targeted with a moiety to the cancer cells, and when they are irradiated with light or they are shown upon with the light, they are able to produce photothermal effect, and thereby they are causing reduction in the tumor size of the mouse model that they have used. So this is another interesting property of these carbon dots. And obviously, not all carbon dot will show this property. People have to actually engineer the carbon dots using doping and surface functionalization effects. So that's what was the take-home message from this study. Then carbon dots, they because of their several functional groups present on the surface, they could be easily conjugated with different nanomaterials as well. So we can form conjugates of or nanocomposites of carbon dots. For example, in this study, what they did, they produced mesoporous silica nanoparticles. These are very highly porous silica nanoparticles, and these silica nanoparticles, they had amine functional group over their surface. And these amine functional group, they attached with folic acid, which also act as a receptor for various cancer cells, and they did a hydrothermal treatment and converted some of the folic acid which is attached on the surface of this mesoporous silica nanoparticle with, they converted that to carbon dots which were fluorescent, when they are excited with 418 nanometer, they produced yellow color emission of 570 nanometer. So now once fluorescence has come because of these carbon dots, they loaded this mesoporous silica nanoparticle. As I already said, that these are very porous silica nanoparticles. They loaded this with doxorubicin, which is a anti-cancer drug, and they treated the cells with this composite. So because of the carbon dots, now the release profile of the doxorubicin could be tracked in real time. So this carbon, because the pH of the cancer cells is lower, therefore this doxorubicin is released into the cancer cell lumen, and the release profile or the spatiotemporal profile of the drug release can be easily studied because of this carbon dots. The carbon dots, as I have previously given an example, that if a carbon dot is PEI covered, that is, it is covered with a positively charged polyethylene polymer, it is able to translocate through the plasma membrane into the cell cytoplasm. So, and since it is positively charged, it could take up several DNA, I would call it DNA or RNA. In this case, it was siRNA, that small interfering RNA that they have utilized. Because of the negatively charged background of this DNA or RNA, this carbon dot, which is covered with PEI, can easily conjugate with it electrostatically. It can conjugate with it, and it can translocate into the cell and release the siRNA there because of the pH difference. Because the carbon dot, the surface is very polar, functional groups are there. So any variation in the pH will cause reversal of these electrostatic interaction. So, with the release of this siRNA, now it could be targeted to a particular mRNA and it could knock down any specific gene. So this study they carried out with using GFP, green fluorescent protein gene, and they could clearly show that when carbon dot is combined with siRNA, it knocks down that particular gene. And they have also attached a Cy5 fluorescent dye to actually study the release profile of the siRNA as well. So so it now we have a dual mode imaging where we can clearly see the carbon dot has released into a lumen, and then siRNA goes to the nucleus and it knocks down the gene which is present. So as the carbon dots, they they are very much responsive to the surface or to the immediate vicinity. Now, so therefore they are able to interact with several metal ions. For example, people have shown that carbon dioxide can directly interact with copper or iron metal ions, and because of this interaction, the electronic property of the carbon dot changes and it becomes no longer fluorescence. So it quenches basically. So those type of sensors, metal ion sensors, which is based on the quenching of the carbon dots, they are called as turn off type sensors. Now in this particular interesting in this study, what was interesting is that they produced carbon dots which is doped with nitrogen and sulfur. So if the sulfur atoms which are present on the surface of the carbon dots, they can now interact with gold nanoparticles because of the gold thiol bond, which is very stable and very well known. So now gold, they have this plasmonic property of quenching. Gold nanoparticles, they actually behaves as a very efficient quencher of fluorescence. So what happens when these carbon dots are excited with a light, the emission which is coming out from the carbon dot is immediately absorbed by the gold nanoparticles through a phenomena called as Foster resonance energy transfer. I guess you all know. So this happens at the surface of the gold nanoparticles. And when glutathione, which is a marker for several for several diseases, uh, so it is basically an antioxidant agent which is present in normal cells, and its levels usually goes up in cancerous cells. So this glutathione, it has a free sulfhydryl group. So this glutathione, when it comes in contact with the gold nanoparticle, it immediately replaces the carbon dots from the gold nanoparticle surface. And because of this replacement, the carbon dots now become fluorescent. So what we get is a turn on type biosensor in this case. Then another very interesting application which is being recently explored in case of carbon dot is something called as BRET sensor. The BRET, similar to FRET, actually stands for bioluminescence resonance energy transfer. In this case, what happens is that a carbon dot is conjugated with luciferase enzyme. So this luciferase enzyme, it when encounters its substrate and some co-substrate, for example, if someone is using firefly luciferase, it uses ATP as co-substrate and luciferin as a substrate. So when it encounters its substrate and co-substrate, it produces light, bioluminescent light. This bioluminescent light is then now absorbed by the carbon dot and it produces its own emission. That carbon dot emission now can be in this, for example, in this study, what they did, they formed this complex of carbon dot with luciferase enzyme, and when luciferase light is produced, it is absorbed by the carbon dots, and that light is now again absorbed by a nearby photosensitizer, which produces reactive oxygen species on being excited. So in this case, they studied a anti-cancer role for this particular application. So even if we don't have any photosensitizer here, then also we can use this BRET emission for biosensing purposes. So there is another application for this BRET is biosensing applications. In like up till now, all the studies that I have quoted is for basically cellular studies, the mammalian cell studies, for anti-cancer studies. Now there is also now examples coming out for antimicrobial activity of carbon dot, where the surface functional group of carbon dot can be easily combined with various polymers. For example, in this case, it is combined with activated dextran polymer, which is activated and generated a aldehyde group. The amine group of the carbon dots can easily interact with aldehyde groups and they forms a hydrogen. And now when these carbon dots are excited with light, for example, here the excitation is shown, they produces photodynamic effect like a reactive oxygen species, and this reactive oxygen species in turn can kill the bacteria which is present in the immediate vicinity. So such kind of wound healing gel or hydrogel is now being produced using carbon dot as the antimicrobial reagent in this case. So that's all from my side. Thank you for listening patiently.
Thank you so much, sir, for this for enlightening us on carbon quantum dots. It was really a wonderful session with you. So now I would like to proceed to the Q&A session where I would read out the questions from the chat box, and I would request Dr. Girish to please enlighten us on the answers to those questions. Now, also, like if anyone has any doubt in particular, they can unmute and also ask sir. And before we get started with the Q&A session, I would like to add that the feedback form is open for 1 hour until now. And please go and form the feedback form if you want to get your certificates. And we would not entertain any pleas after 1 hour. So please go and fill the feedback form. So now moving on to the Q&A session. The first question that we have here is that, is there any difference between carbon quantum dots and carbon nanoparticles? And which of these is better for targeted drug delivery?
Carbon nanoparticle, as in, I mean, carbon nanoparticles and carbon dots are mostly similar. I mean, if we are talking about if carbon nanoparticles is fluorescent or not, so carbon, if it is a fluorescent, it is actually carbon dots only. So people have actually they have used very interchangeably these terms, carbon dots, some call it carbon quantum dots. The only difference, the major difference between carbon dots, carbon nanodiamonds, and graphene quantum dots, these three are the fluorescent allotropes, nanoallotropes of carbon. So the basic difference between each of them lies as such. Like graphene quantum dots, these are specifically derived from graphenes. They are also of similar size between 2 to 20 nanometer, and but instead of a spherical particle, the graphene quantum dots are usually nanosheets of graphene, which are in 2 to 20 nanometer dimension. So few layer of graphene nanosheets. So that's the difference between a graphene quantum dot and a carbon dots. In case of carbon nanodiamonds, the core of the carbon nanodiamonds is usually sp3 hybridized. And as we know from the example of diamond, which is very hard to make, right? Diamond, it forms at very high temperature and very high pressure. So that sp3 hybridization of carbon is very difficult to achieve. So similarly, in case of nanodiamonds, usually nanodiamonds are formed in very high combustion reactions. For example, some explosion is there or arc discharge reactions are there, then only nanodiamonds are formed. So majority of the core of the nanodiamonds is basically sp3 hybridized. And in terms of fluorescence, nanodiamonds are usually less fluorescent as compared with carbon dots. And as far as I know, the control of fluorescence over nanodiamond has not been achieved yet. So people have actually moved on from carbon nanodiamonds to carbon dots. In case of carbon dots, the carbon dot structure, uh, people consider it as having a graphitic core, having both sp2 and sp3 hybridized carbons, and then an amorphous layer, which is generally consisting of heteroatoms like nitrogen, oxygen, various functional groups are present. So that's the difference. So carbon nanoparticle, I would call it a very general term. One has to go beyond that to define exactly out of these three fluorescent carbon nanoallotropes what we are talking about. I hope that clears it. Or
No, sir, I think that was absolutely fantastic. And moving on to the next question. The next question is that, what are the factors affecting plasmon response of carbon quantum dots?
plasmon response of carbon well, plasmonic response of carbon dots is actually not well studied. I mean, see, there is a difference between fluorescence response and the plasmonic response. So, I mean, for example, gold nanoparticles, those are plasmonic materials because they have a particular absorption of light in the visible region, right? And it has a free, I mean, surface free oscillating electrons over the surface of gold, silver, as well as copper. Whereas in case of carbon dots, there is no such free electrons present at the surface of the carbon dots. If they are talking about fluorescence response of the carbon dots, the fluorescence response is basically depending, as I mentioned, three factors before. It basically depends on the surface polarity, that is, what are the surface functional groups of present at the surface. Like two studies that I have shown before, that the fluorescence response depends on the size of the carbon dots because of the quantum confinement effect. So lower the size, the bluer will be the emission. That means as size reduces, the emission shifts to blue shifts, whereas as size increases, emission red shifts. Whereas in case of surface response, if the number of oxygen electron rich atoms, heteroatoms, for example, sulfur is there, then there are oxygen is there, nitrogen is there. When the number of such electron rich atoms increases, that is the oxidation state increases, then the band gap also changes, and that takes the emission to a towards a red shift region, I mean red region.
Moving on to the next question, it is that can we use carbon quantum dots in gene therapy as gene carrier instead of viral vectors?
Yeah, definitely. For example, I just quoted one example. There are several other studies where they have used carbon dots or carbon quantum dot, you call it, for gene therapy, where the carbon, because of the surface charge of the carbon quantum dots, if it is a positively charged, then it could take up a large amount of DNA with it, or DNA or any RNA, because both of them are negatively charged backbone, because of the that phosphodiester bond. So the backbone of the DNA and RNA is negatively charged. So positively charged carbon dots, they take up these DNA molecules and could translocate through the plasma membrane. The another important factor in this case is that it basically size is very smaller of carbon dots, so they could easily translocate through the pores of the membrane. So it can absolutely used as gene therapy, and not only it will be helpful for translocating the gene across the plasma membrane, but we can actually also track the movement of the carbon dot because of its fluorescence. So that's the additional advantage that we get out of it.
So before we discuss the last question, I would like Abhijit to unmute his mic as he has been raising his hand since long. Abhijit, please move forward with your doubt.
Thank you, Priyanshu. Sir, I actually have a doubt. Do carbon quantum dots have any penetrating power?
Carbon quantum dot have penetrating power in cells, you mean to say?
Yes, sir. As in,
They
could they penetrate individual cells?
they could penetrate individual cells based on the surface charge. That is what actually important is.
Oh,
Carbon dots, they have, if they have positively charged, so our so in our cells, all the cells, they are actually phospholipids, right, in the membrane. So this, our cells are
yes, sir.
basically negatively charged. So when we encounter any positively charged nanoparticle, for example, carbon dots, it could translocate across the cells easily, because size is very low.
Sir, if so, sir, then suppose carbon dots were able to penetrate a cell, so it could also apply to tumor cells as well. So, sir,
Yes,
then
definitely.
can't we excite the carbon dots and lead to their selective destruction of
Yes,
tumor cells?
yes, that can be done, but see, if we are not attaching in a targeting molecule, right? So if we have a population of healthy cells as well as tumor cells, and we are just incubating these cells with carbon dots, positively charged carbon dots. Now cells, these carbon dots will have an equal propensity to go inside every kind of cells, be it healthy or be it tumorous cells. In some cases, if we consider it as a tumor tissue rather than cells, then there will be some propensity to go more into tumor tissue because of the porosity of this tumor tissues. You know about vascular porosity of tumor tissues. So in that case, that's the case. But if we are not functionalizing of a carbon dot without anything, without any targeting molecule, then it could penetrate both healthy as well as tumor cells. And when irradiated, it will definitely produce the photodynamic or photothermal effect, and it will definitely produce cellular toxicity in both the cells.
Okay,
You
sir.
need to have some targeting molecule which will accumulate large number of carbon quantum dots in a vicinity of a particular cell. That is the purpose of basically targeting molecules.
Okay, sir. Thank you.
And next, Soumya Mathur, you may proceed with your question.
Good evening, sir.
Good evening, Soumya.
Sir, I was wondering that all of these are very lab experiments. Is there any way we could study the properties of quantum dots computationally, like maybe using DFT or something of that sort?
Yes, there are several computational studies people have done. In fact, this functional group modification that I am talking about, the surface functionalization and surface modification, people have actually done it computationally first to see what will be the changes in the electronic property of the carbon dot and how it will affect the fluorescence of those carbon dots. And then they have actually went ahead and did it experimentally. So so people are using it for computational studies as well.
So, if you would you recommend DFT? Because I spent some time in the past 6 months studying DFT.
Actually, I'm not from computational background actually, so I won't
Okay.
be able to comment on exactly which model to take. But yes,
Mhm.
there are several papers that you could follow, and or I could connect you with someone who is working computationally over all these things.
So because there is the fluorescence as a property. Are there any other factors that define how
That define
effective
carbon
quantum?
dots.
Yes, sir.
Carbon dots, they are usually used for fluorescence as well. I mean, the majority of their application is based on fluorescence. Since they are very small, so their molecular arrangement that we try to do it for, in case of, let's say, graphene, we use it for electronic property, right? So that kind of control using carbon dot, they have not utilized much. I mean, they have used it for it's for fluorescent property or photodynamic therapy or photothermal therapy. These are the major applications for carbon dots from that perspective. Usually associated with the optical properties. I mean to say.
Okay, sir. Got it. Thank you.
People they see all the nanoparticles, they have very high surface area, right? And carbon dots, they have very rich functional group over their surface. So there are studies where they have used it carbon dots for adsorption purposes, like they could adsorb some antibiotic or some pesticides from the waste effluents, but nothing sort of engineering aspect that I have I mean obviously they are working for LED development also, light emitting diode also. So, but my knowledge in those domains is limited, little limited.
Okay, sir. Thank you.
Thank you, sir. That was a really wonderful Q&A session with you, and I hope that it cleared all of our doubts regarding this webinar. So now, as every beautiful thing in this world has to come to an end, and so has to this webinar. So now I would request the treasurer of Genesys, Parthipan Bhaiya, to give the vote of thanks. Parthipan Bhaiya, you may take over now.
Yeah, Priyansha, am I audible?
Yes,
Yes,
yes.
audible.
Yeah. So yeah, a pleasant evening to one and all present over here. Myself Parthipan, a third year student of Life Sciences Department and treasurer of Genesys, would like to use this golden opportunity to deliver a vote of thanks to all dignitaries present over here. First of all, I would like to thank Dr. Girish Chandra Mohanta sir for sharing his knowledge and experience on biomedical applications of carbon quantum dots. We are very fortunate to have you here, sir, and thanks for sharing your time despite of your busy schedule. And I hope you had a great time as well with us. Next, I would like to thank the Biotechnology and Medical Engineering Department professors for your constant presence and support. I would also like to thank Priyanshu for smoothly hosting the webinar the entire team of Genesys, students, and our beloved audience from various institutes for making this webinar a grand success. Hope you all enjoy the event, and I believe the knowledge you gain today will help you in some aspects of your future endeavors. The entire team of Genesys wishes you all a very good luck, and yeah, we will all see you in our closing ceremony. Until then, yeah, stay safe and goodbye. Thank you, one and all.